Hello, and welcome to Implementation of the SoloVPE Technology for Protein Concentration Measurement of Monoclonal Antibodies, presented by American Pharmaceutical Review and sponsored by C Technologies, a Repligen company. My name is Mike Auerbach. I'm the Editor-in-Chief of American Pharmaceutical Review, and I'll be the moderator during today's event. In this webinar, you will learn the benefits of implementing the SoloVPE technology for protein concentration analysis in a quality control laboratory, the challenges experienced during method implementation and validation, particularly for high protein concentration samples, and the method characteristics evaluated during validation. In addition, this event will hold a live question and answer session at the end of the presentation. To ask a question, click on the Ask a Question tab on the right side of your screen. Please take note the right side of your screen also features an overview of this webinar and more information regarding our speaker. If you have a technical question during the event, click the Test Your Connection button at the bottom of your screen. From there, you can access additional webinar support. Finally, we encourage you to use the social media widgets beneath the webinar to share with your friends and colleagues. And now, allow me to introduce today's speaker. Maria Chomova completed her undergraduate studies at the College of Engineering at Cornell University. She currently works as a quality control supervisor at Regeneron Pharmaceuticals, managing the optimization and validation of analytical methods as well as implementation and transfer of methods to QC release laboratories. Maria, welcome to the webinar.
Thank you, Mike, for that introduction, and welcome everyone to today's webinar. First, we'll overview some of the topics that we'll discuss today. Starting off, we'll talk about the importance of protein concentration in monoclonal antibody production, some of the challenges and limitations we see with the traditional UV methods, and then we'll go into the implementation of the SoloVPE technology in GMP quality control laboratories. Specifically, we'll review the principles and the benefits of the SoloVPE, our implementation experience at Regeneron, the method characteristics evaluated during validation of the method, some of the challenges we've seen with high concentration products, and additional post-implementation improvements that we've made at Regeneron. And then we'll end the webinar summarizing our discussions today. So we've all heard of monoclonal antibodies in terms of the immune system using them to identify and neutralize foreign objects. But we can also manufacture these antibodies to be used as therapeutic agents to treat disease. Monoclonal antibodies are proteins produced by a single cell line and designed to bind a specific antigen in the body. This allows us to use them to target and regulate specific cell processes, or even to flag cancer cells for destruction. Now, producing these monoclonal antibodies involves a very complex manufacturing process. To simply explain this process, it is composed of an upstream cell culture process, where a culture of cells is grown under controlled conditions to produce the antibody of interest. This is then followed by a downstream process where the antibody is purified through a series of unit operations, typically consisting of resin-packed columns that are designed to remove impurities that have been introduced or generated by the cell culture process. And finally, the purified antibody is formulated, dispensed, and packaged for administration to the clinic. So now we know what a monoclonal antibody is and why we manufacture it, but why is protein concentration important to us in this process? Well, concentration is important in the manufacturing process because it is used to calculate the optimum protein load for each unit operation. It is very important to find a correct balance of analyte and resin contact in order to gain maximum yield of the product. Without knowing the protein concentration, we risk overloading the resin, which can lead to loss of product, or we could also risk underloading the resin, which leads to inefficiency as we require an increased number of cycles to complete purification of the antibody, and therefore higher resin usage and even shorter resin lifetime. Concentration is also important in the final product. The concentration is critical when assessing the pharmacokinetics of the monoclonal antibody and selecting an effective dosing regimen for the patient. Oftentimes, the final product can be administered in different doses during a clinical trial. If the product concentration is not accurate, we risk misdosing a patient, which can then lead to inaccurate study results and potentially health complications. Most importantly, this is a regulatory requirement. In order to demonstrate product quality, we are required to demonstrate proper protein concentration, protein identity, and purity. Protein concentration is also important during quality control testing of the product. Analytical methods are often concentration dependent as they require samples and standards to be diluted at an equivalent amount for analysis. Inaccurate sample dilutions could lead to invalid test results or inaccurate results, which could in turn compromise the integrity and the safety of the product. In addition, it is important to ensure that testing occurs within the validated method ranges. This ensures that we have systematic controls in place to identify out of specification or atypical product. If we use the incorrect concentration, we risk operating outside of these validated controls. Certain assays can also be extremely sensitive to concentration. Potency, for example, tests the activity of the monoclonal antibody relative to a known amount of antigen that is present in a solution. Other methods test for process impurities and report these impurities in the final product relative to the patient dose. This is to ensure that acceptable daily exposure limits are not exceeded. Now we know why concentration is important, but how do we measure it, and what are some challenges and limitations experienced with the traditional methods? Traditionally, monoclonal antibody concentration is measured using UV spectroscopy. As we mentioned earlier, antibodies are proteins, and proteins are composed of a chain of amino acids. Some of these amino acids, such as tryptophan and tyrosine, contain aromatic rings, which exhibit strong UV light absorption at 280 nanometers. Because of these amino acids, monoclonal antibodies also absorb UV light most strongly at 280 nanometers. The number of aromatic rings in a particular antibody determine how strongly it absorbs light, making this an intrinsic property that is represented by the extinction coefficient of the antibody. This characteristic allows us to measure antibody concentration in solution using UV spectroscopy and the Beer-Lambert law. The Beer-Lambert law states that absorbance at a specific wavelength is proportional to the extinction coefficient of the analyte at that wavelength times the path length of light emitted times the concentration of the analyte. So, in principle, when we know the extinction coefficient of the analyte at a specific wavelength and path length is kept constant, there is a linear relationship between absorbance at that wavelength and concentration of the sample. In other words, a higher concentration of antibody in solution would translate to a higher UV absorbance at 280 nanometers. As we can see on the spectrum in this slide, which compares a protein sample in the red trace to a water and buffer solution that do not contain any protein and therefore do not show any absorbance at 280 nanometers. So now we understand the principles of UV spectroscopy, but as with most instruments, there are limitations. It is difficult to obtain high-resolution measurements without increasing measurement times or producing very expensive and difficult-to-manufacture equipment. As a result, we run into challenges with traditional UV spectrometers, where measurements start to diverge from the Beer-Lambert law. This is particularly due to stray light and absorbance saturation. Stray light is light that reaches the detector but is outside the spectral region isolated for measurements. It can originate inside the instrument optics or outside the instrument from light leaks. As seen in this graph on the slide, the more stray light that is transmitted, the sooner the instrument measurements start to diverge from the linear correlation of absorbance and concentration. In addition, measurements hit an absorbance limit, or saturation. Due to these instrument limitations, samples are often required dilution to ensure that measurements remain within the linear range of the instrument. This is particularly difficult for high concentration samples, as it leads to larger dilution factors and often the need to perform multiple dilutions in series. This poses higher risk for error due to analyst and equipment variability. So having to dilute samples typically leads to a very laborious process, especially in the quality control space. Quality control laboratories operate under stringent conditions to ensure that product tested and released into the clinic meets all regulatory expectations and is safe for administration to the patient. A total protein method in a QC lab could incorporate the workflow shown on this slide. Starting out, a typical QC workflow would involve a daily instrument check, which ensures that the instrument is set up to perform as expected. This would then be followed by the collection of a background measurement to account for any stray light limitations. This would be followed by the collection of a blank measurement. Typically, the blank is the diluent that is used to prepare the samples. This is done to ensure that minimal diluent interference is observed or to correct for the interference that is observed. This is then followed by the measurement of an assay control, which can be either a purchased or an in-house prepared standard that is qualified and expected to produce measurements within a specified range. This is then followed by sample dilution. In QC, samples are typically diluted in triplicate in order to mitigate variability from analyst technique. Depending on the target concentrations, sample preparations may also require serial dilutions to avoid pipetting small volumes. Proteinaceous material is especially highly viscous, which can lead to variability and inaccuracy when pipetting small volumes. On top of diluting in triplicate, QC methods may require daily pipette verifications to ensure the pipettes used for the dilutions are accurate and that the analyst is able to precisely deliver the required volume. Following dilution, each sample preparation is measured on the UV spectrometer. Once all data is collected, scatter correction and concentration are calculated manually for each measurement, as well as for the average of all preparations. Because manual calculations pose more risk for error, additional data review steps are often required, consuming even more time and resources in the QC laboratory. Finally, the optimal concentration ranges are implemented for each product based on the extinction coefficient of the product. This makes it very difficult to apply and implement a platform approach of total protein measurement within the QC lab. So given some of the limitations discussed with the traditional method, next we'll overview some benefits and challenges experienced with the implementation of the SoloVPE technology. Similar to traditional UV methods, the slope spectroscopy technique also employs the Beer-Lambert law. However, instead of keeping path length constant and varying the concentration, the SoloVPE instrument is able to vary the path length while keeping the concentration constant. In other words, if we know the extinction coefficient of the analyte at a specific wavelength and we keep concentration constant, there is a linear correlation between the absorbance at that wavelength and the path length of the instrument. Some of the key features of the SoloVPE are that path length selection is controlled and optimized by the instrument. The instrument generates 5 to 10 absorbance measurements at different path lengths within the linear range. In addition, the software calculates the sample concentration using the generated slope, the user-supplied extinction coefficient, and the Beer-Lambert law. These key features of the SoloVPE drastically simplify the workflow of the QC protein concentration method. As we can see on the slide, we can simplify a QC workflow down to 4 major steps. A daily instrument check would still be performed to ensure that the instrument is set up and operating as intended. An assay control measurement would still be collected as a secondary layer of control to ensure that we meet the stringent QC regulations. However, sample dilution would no longer be required as sample concentration is the constant and does not have to fall within the linear range of the instrument. In this case, path length is the variable used to generate the linear relationship to absorbance, and path length is controlled by the instrument. In addition to not requiring sample dilution, a single sample measurement can be implemented as method variability is dependent on the instrument, and that is already controlled through our daily checks and assay control measurements. In other words, we are not introducing any more external variability factors such as dilution variability from the analyst or the pipette. And finally, the software automatically generates and reports a sample concentration, removing the need for manual calculations and additional data review steps. Just through the shortened method workflow, we can see many of the benefits of SoloVPE over traditional UV methods. To compare them directly, both methods would require a daily instrument check. However, the SoloVPE contains an internal daily check that automatically populates a pass or fail result, whereas traditional UV methods typically require calibrated standards. Both methods would also require an assay control to ensure that QC regulations are met. However, with SoloVPE, there is an option to purchase a confirmed standard already supplied by the vendor that comes with a certified value, whereas for traditional methods, we typically have to prepare in-house controls and qualify them ourselves. The SoloVPE would not require any sample dilution, whereas traditional UV typically requires dilutions in triplicate. SoloVPE would not require a blank measurement as we're not diluting the samples and do not have to test the diluent, whereas traditional UV would require this blank measurement to ensure minimal background interference. The SoloVPE automatically reports the sample concentration, whereas traditional UV requires manual calculations. And finally, the sample used for SoloVPE measurements is often recoverable, allowing us to use it for other QC assays, whereas traditional UV requires sample manipulation, which makes it difficult to recover the sample and use it for other purposes. In summary, just by removing sample dilution and automating result reporting, we're able to save time in the quality control laboratories, approximately 45 minutes per sample if you factor in sample dilutions, measuring every preparation, manually calculating results, and having a second verifier to review those calculations. We're also able to lower the invalid rate as we are eliminating any variability from analysts as well as pipettes. By saving time and lowering the invalid rate, we're able to implement a much higher throughput method within the QC laboratory. At Regeneron, we have implemented the SoloVPE technology in multiple areas of the business, some of which include our research and development sector, where we work on product discovery and formulation development, our process areas, where operators are able to take immediate on-the-floor measurements to continue manufacturing operations, our quality control laboratories, which focus on product quality, and even some external contract manufacturing organizations, where we're able to easily transfer the SoloVPE method for other uses such as placebo identification prior to filling operations. To go more in depth in quality control, we have implemented protein concentration analysis for in-process, release, and stability testing. In-process samples are tested to ensure the process controls are maintained during the manufacturing of the monoclonal antibody. And release and stability samples are tested for specifications to ensure product quality and regulatory compliance. In QC, we have validated the SoloVPE method for a range of concentrations, all the way from 0.1 milligrams per milliliter to 279 milligrams per milliliter. We have also been able to validate this method for a range of extinction coefficients and an array of product excipients. In addition to testing for product quality, we have implemented SoloVPE for mid-assay concentration measurements. Various analytical techniques require confirmation of sample concentration during the assay, such as after a filtration step or after a reconstitution step. SoloVPE makes these steps more efficient as operators do not have to stop mid-assay to dilute samples and manually perform calculations. Furthermore, the SoloVPE has been implemented for cleaning studies where we ensure the product has been cleared of the process. So overall, SoloVPE seems like a great solution to the traditional UV challenges we experience, but some method characteristics must still be evaluated in detail, especially when implementing and validating this technology in the QC space for the first time. Example characteristics that we focus on at Regeneron include matrix specificity, which is very important to ensure that our sample matrix components are not interfering with the protein detection. Scatter correction, which is important to determine if scatter correction is even appropriate for each product that we analyze. Linearity and range of the method, which is absolutely important in our case where we test low concentration in process pools all the way through very highly concentrated final product. Method accuracy, which just ensures that the instrument is performing as expected and outputting measurements that we expect for our samples. And the most important is repeatability and precision, which ensures that the method is consistently able to produce results, especially through a range of different analysts and instruments. So shown on this slide is some of the precision data generated at Regeneron for our monoclonal antibody products. This data specifically was generated using 4 different analysts and 3 different instruments across a total of 8 analyses. Each analysis consisted of triplicate measurements just for the purposes of method validation, therefore producing a total of 24 measurements for this one sample. It can be seen that across all 24 measurements, we are able to produce consistently accurate results compared to our target concentration of 76.8 milligrams per milliliter. In addition, all measurements across all of the analysts and instruments demonstrated low variability. We were able to achieve a standard deviation of only 1.1 milligram per milliliter and an overall percent RSD of 1.4%. Precision was also evaluated across 4 analysts and 3 instruments for lower concentration samples. On this slide, we're showing 2 samples, one with a target concentration of around 1 milligram per milliliter, and a second sample with a target concentration of 49.7 milligrams per milliliter. So again, across all 24 measurements, we can see that we produce very accurate results compared to the target and very low variability, with a standard deviation of 0.02 mg per mil for the 1 mg per mil sample, along with a 2.3% RSD, and a standard deviation of only 0.6 mg per mil for the 49 mg per mil sample, and an RSD of 1.1%. So overall, the precision data we observed for low to mid concentration samples was well controlled and very easy to implement into the QC laboratory. However, at Regeneron, we ran into some challenges when implementing the SoloVPE for higher concentration samples above the range of 120 milligrams per mil. As mentioned before, the SoloVPE instrument generates 5 to 10 absorbance measurements at different path lengths. For the high concentration samples, this 10-point analysis sometimes exceeds the linear range of the instrument. As shown in this graph, the UV absorbance values above a certain path length begin to taper out and diverge from the linear range of the Beer-Lambert law. This leads to large variability in sample measurements, especially across multiple analysts and multiple instruments, and sometimes produce artificially low concentrations for our samples. However, as we can see in the graph, there is still a linear range at lower path lengths that can be used for these concentration measurements. We're able to correct this issue by limiting the instrument analysis to only 5 or 6 points, where the instrument is able to choose the smallest path lengths when measuring high concentration samples and the largest path lengths when measuring low concentration samples. This ensures that all measurements remain within the linear range. So to share some of our data at Regeneron, we analyzed 2 high concentration products using different techniques. The top chart here displays 6 measurements of a product with a target concentration of 210 milligrams per mil using a 10 path length measurement. It can be noted that at the maximum path length, the absorbance values begin to exceed 2 absorbance units. This is where the method begins to diverge from the Beer-Lambert law, as we saw on the previous slide. In addition, the variability we observed between the 6 measurements for the sample is very high, at 2.5% RSD and 4.8 milligrams per milliliter standard deviation. Note that all 6 measurements were taken by the same analyst on the same instrument, so that percent RSD of 2.5 is considered very high. The bottom chart, on the other hand, displays 6 measurements of a product with a target concentration greater than 180 mg per mil. These measurements use only 5 path length measurements on the instrument. It can be noted here that the instrument limits the maximum path length it uses as compared to the 10-point method above, so that maximum absorbance is kept within the target range of 1 absorbance unit. This measurement is able to produce much higher R-squared values and much lower variability across our 6 measurements. We're able to achieve a 0.9% RSD and a 1.8 mg per mil standard deviation. Another product we measured with a target concentration of 220 mg per mil was analyzed using 3 different techniques. All the data here was collected across a range of 10 different analysts in order to add some more variability. The top row shows our data using 10-point measurements with no sample dilution, followed by a 5-point measurement, again no sample dilution, but this time not using scatter correction, and finally a 5-point measurement, no dilution, with applied scatter correction at 320 nanometers. So we can see that the 10-point method in this case produced the highest delta concentrations across the range, along with the highest RSD and the higher standard deviation. The 2 5-point methods, on the other hand, were able to produce a much tighter range across all measurements and a much lower overall variability at only 1.2% RSD and 2.7 mg per mil standard deviation. It can also be noted here that scatter correction did not impact our final results for this product, as both measurements resulted in the same average reported concentration. This is where we go back to having to assess scatter correction for each product to determine if it's appropriate. So great, we're able to mitigate the high concentration challenges. But our work doesn't stop there. We continue to implement method improvements to help decrease variability and burden on the QC operators. Here are some other examples of improvements we've implemented. At one point, we began to observe high invalid rates due to improper manual fibrette installation. This led to a lot of broken fibrettes, failed R-squared criteria, and having to invalidate and repeat testing. To mitigate this issue, we were able to install the quick-set coupler attachment to all of our instruments. This ensured that operators could correctly position the fibrette with just one click and not have to worry about failed runs. Another issue we started to observe was variability due to analyst techniques. In order to mitigate this issue, we implemented a pre-analysis check using the vendor-supplied confirmed Phillips reference standard. This allowed us to improve our control over the assay as well as over the analyst techniques. So in summary, let's review the key topics we discussed today. Protein concentration is a critical attribute and a regulatory requirement for the manufacture of monoclonal antibodies. As we discussed, concentration is used both in the upstream and downstream manufacturing processes. It's used to determine safe and effective patient dosing, and it is used in analytical methods for product release and stability. Implementing the SoloVPE technology saves significant time compared to traditional UV methods, especially in the quality control space. By eliminating sample dilutions, we're able to reduce on-bench time and human error. By automating the sample reporting, we're able to reduce documentation time and errors, as well as the need to have a second verifier. Overall, this results in a high throughput assay. For example, in our QC labs, we typically test and release an average of 155 samples per week. This saves us about 9 hours per day using the SoloVPE as compared to our traditional UV method. Furthermore, the SoloVPE method is easily transferred to other labs and sites across the business, as we don't have to worry about training the analysts to properly dilute samples and calculate concentrations. Overall, as a final note, when implementing the SoloVPE, especially in the quality control space, it's important to consider the range of protein concentrations that will be evaluated and tested for each product. This ensures that we implement optimal instrument parameters right off the bat. So in closing, I would like to acknowledge my colleagues at Regeneron and the team at Repligen for all their work in implementing the SoloVPE technology. Thank you for your time, everyone, and now we will open it up to questions.
Thanks, Maria, for your excellent presentation. I'd like to remind everyone watching that you can submit any questions you have for Maria in the Q&A box on your screen, and we'll get to as many as we can. So let's take a look at our questions. And Maria, our first question is, understanding that this presentation is limited to mAbs, have you at Regeneron tested other protein modalities on SoloVPE with varying extinction coefficients?
Thanks, Mike, for the question. At Regeneron specifically, we currently focus only on monoclonal antibodies. However, there are documents out there of other modalities being tested using this technology, and I'm sure Repligen would be able to provide more information.
Terrific, thank you. Okay, let's look at our next question. Have you established the acceptable path length ranges based on high concentration and low concentration products within your SOPs? If the samples don't fall within the chosen path length range, do you deem the assay invalid?
That is a great question. So we actually evaluate each product individually for the optimal path length ranges during method validation, and then we implement those methods into the release testing laboratories. We have other procedures in place within our testing SOPs that would invalidate assays if they're not performing within those validated method ranges.
Terrific. Thanks, Maria. Let's move on to our next question. How does target protein concentration determine for slides 14, 15, and 17? And how does variability determine between different uv-vis techniques versus SoloVPE?
That is a great question as well. So our target concentrations are actually determined in the laboratory while we're formulating our product. They are based on starting off with high concentration samples and then diluting them down with our formulation excipients. Initially, during implementation of the SoloVPE, we do compare the two techniques by analyzing the same samples using both instruments.
Okay, excellent. Let's move on to our next question. Is it possible to measure sample turbidity with SoloVPE?
I could take this one. This is Joe Ferriolo from Repligen. The work has not yet been explored for that application, so it's not a no. We just haven't put the time in for that. To answer the other question that came up as far as additional extinction coefficients, certainly the device is already being used for multiple gene therapy applications, such as oligonucleotides, plasmid purity, empty full analysis for AAV material, which all have very different extinction coefficients compared to a protein.
Excellent. Thank you very much. Let's move on to our next question. How long does it usually take for the SoloVPE system to be installed and validated for QC work?
So that really depends on the company's procedures and their available resources. Typically, at least for first-time implementation of the technology in the QC lab, it could take up to a year. That includes acquiring the instrument, setting it up, qualifying it, setting written procedures in place to guide the operators, and then also validating the methods and training the operators. So any amount of time up to a year.
Mm-hmm. Terrific. Thank you. Let's move on to our next question. Do the quickset couplers have an established shelf life, and how often do you have to change them?
They are changed annually during the preventative maintenance service. You will also have spares included with every system if needed in the interim.
Excellent. Thanks, Joe. Let's move on to our next question. Can you please talk about the validation process and validation support that Repligen provided?
Of course, I can take that on the Regeneron side. Repligen has been very hands-on with us and helping us implement and validate the technology. We still continue to collaborate closely, and they're very good at assisting with troubleshooting as well as method optimization. And then we also have annual service appointments set in place. Like Joe mentioned, those couplers are replaced yearly, but we also have some other services provided by Repligen at a yearly basis.
Mm-hmm. Excellent. Thanks, Maria. Our next question is, you mentioned that other departments are also using the SoloVPE. What type of implementation was needed, and what type of collaboration happened to facilitate adoption in other departments?
Yeah, another great question. Obviously, in research and development environments, it's a bit easier to implement technology like this as we don't have as many regulations and as many written procedures in place, but we do still collaborate very closely between our R&D and our QC and manufacturing departments to make sure that we're aligned across the board on our best practices. We oftentimes will share target and expected concentrations between groups as we transfer material or transfer other types of methods across sites. So there's continual collaboration, continual communication, just to make sure that we're all aligned across the board and across our business.
Excellent. Thank you. Let's look at our next question. Regarding method transfers to external CMOs, was the process easier due to the technique of slope spectroscopy compared to traditional UV?
Yes, absolutely. It is much faster to transfer the slope spectroscopies. There's a lot less analyst preparation, a lot less calculation. So it's faster to train operators to use this technology, and it's also easier on our end to troubleshoot any issues that they have, since there's so much detail output by the software itself that can help us understand what goes on with each measurement.
Mm-hmm. Thanks, Maria. Okay, looking at our next question, have you encountered any product that has varied extinction coefficients at different concentrations due to increased intermolecular interactions, especially at a high concentration? If so, how did you address this issue to establish a valid extinction coefficient suitable for the intended concentration range?
That is a very interesting question. We have not encountered anything like this with our current products.
Okay, terrific, thanks. Let's take a look at our next question, and that is, what is the biggest value that you saw after implementing SoloVPE?
A lot of this was mentioned in the presentation, but I would say the biggest value is definitely our time saved on bench, especially in the QC laboratory. Operators are constantly distracted by everything going on around them, and being able to save them on bench time saves the amount of stress on them and the amount of errors that they may be prone to. But another big value is our ability to recover the sample after taking SoloVPE measurements, and this is especially useful to us while performing other analytical techniques, again, as mentioned in the presentation, since we can quickly verify the concentration and continue with our sample analysis.
Mm-hmm. Excellent. Thanks, Maria. Let's take a look at our next question. Did you have to internally establish comparability between traditional uv-vis and SoloVPE before bringing this on for downstream applications in the QC space for release?
Another great question. So we do not have to do this because we typically implement new technology for new products. That way we don't have to run comparability studies, but we do have a lot of data showing this just for our own interest and confidence in the method.
Mm-hmm. Excellent. Thanks again. Our next question is there a plan to implement SoloVPE for all monoclonal antibody products, or is it already implemented?
Yes, our plan is to implement it for all monoclonal antibodies at Regeneron.
Excellent. Thanks again. Let's take a look at our next question. How would you handle the validation of new products? Does every new product or matrix need to go through the validation process?
That is something that, again, probably depends on the product and the company. In a QC environment, we definitely have to ensure that we're meeting all regulatory requirements. So all the validation data produced for the method and any product that is being released into the clinic has to meet those requirements. And that is what we follow when determining how to handle the validation, what method characteristics have to be evaluated, and if this has to be done for every product.
Mm-hmm. Excellent. Thank you very much. I believe that's all of the questions from the audience. I'd like to thank Maria Chamova for sharing her knowledge with us, and also offer a special thank you to Repligen for sponsoring today's event. Please keep a lookout for an email containing a link to view this webinar on demand and to share with your colleagues. Thanks for attending the webinar, and please enjoy the rest of your day.